Okay. Well, I guess for the benefit of people that aren't here, all of you know who I am now, don't you? But yeah, for the benefit of probably not very many people that might be watching it on the internet, I'm Quince. I'm recently moved to the University of Warwick. Actually, I was at Glasgow until a few weeks ago, and today I'm going to give a sort of overview of some work that I've been doing for the last few years in collaboration with various people on statistical modelling of microbial communities. And my motivation for starting this stuff was an observation that's proved quite controversial in microbial community analysis. And this is this idea of enterotypes, and I'll explain what I mean by that in a minute. And that motivated me to come up with a probabilistic model to try and describe these enterotypes, and that was these Dirichlet multinomial mixtures. And then after a while, I sort of realized that modeling structure could be extended to address this question of how to fit Hubble's unified neutral theory of biodiversity. And that's some work I did with Todd Parsons, who's, I think, in Paris at the moment, isn't he? But who attended Todd's talk a few weeks ago? Yeah. So Todd, I'm going to give the sort of data analysis side to that same topic. So Todd showed that you could. I don't think Todd presented data, did he? No, he showed this mathematical connection between Hubble's neutral theory and this thing called the hierarchical Dirichlet process. I'm going to show how you can then use that hierarchical Dirichlet process to fit Hubble's model. Okay. So enterotypes. This has been I don't know if you guys are aware of this, but it's been one of the most controversial topics in microbial community analysis over the last 3 years. I literally watched two professors almost have a fist fight at a conference. The worst thing was I was meant to be the chairman of the. I just sat there. Well, I should have done something. Anyway, the idea of enterotypes is actually that if you take the human gut microbiota, then it falls into 3 distinct groups. And in the original paper, they were very clear about it. They described them as robust clusters, referred to as enterotypes hereafter. So they identified three groups, and those groups were distinguished by the composition of the community. So as one group was dominated by bacteroides, I think that's shown in green here. Another group was dominated by Prevotella, that's shown in red, and then they had a third group which had more ruminococcus. And this was very exciting and very much played up in the press. It was a Nature paper. And the idea was that these things could almost be like blood types, if you think about the consequences of that for medicine, for example, it's quite huge implications. However, so people did additional analysis on the same and further data sets, and things did not appear as clear as they did in the original paper. So Susan Holmes, a statistician at Berkeley, did a lot of work on this. And she showed that these clusters in the original paper are basically not robust to the choice of the distance you use. So they did a distance-based clustering. They basically used something called partitioning around medoids. And it's a bit like k-means clustering, if any of you are aware of clustering algorithms. But it was getting those 3 clusters was very dependent upon the choice of the distance measure you use. And that's the key thing to this type of analysis. I mean, they're not really robust clusters if you try 12 distance measures and only one gives you the distinct clusters. And then there was a reanalysis of a larger data set, the HMP data set that was done by Karen et al. And they got some evidence for two clusters, but not the 3 seen in the original study. But they were still using this partitioning around medoids approach. So consequence of this is actually they published an addendum where they sort of rephrased their article. So they're now saying that they're not distinct clusters, but more as densely populated areas in a space of community configurations. Now, things like partitioning around medoids are not actually a good model to describe that type of structure. The clustering algorithms are looking for distinct clusters. So, partially motivated by this, I mean, I was doing it sort of in parallel to a lot of this discussion, I wanted to come up with a more flexible model, a true probabilistic model that could describe these ideas of diffuse clusters of community types. And that's what I'll explain to you. Now, the starting point for this is the data matrix itself. Okay, so we're going to assume that we've got big N samples, and within those samples we have a total of big S different species. And then the raw data for all these analyses are just the counts of the species. If you're interested, someday I could explain to you the bioinformatics complexities of how to get those counts, but it's not really relevant to this. We just consider this a data matrix. But this data matrix does have some important features. Firstly, it's discrete. Also, it's a variable size. So each sample has a different number of counts associated with it, and that just comes down to the arbitrariness of the sequencing library preparation. It's also fairly sparse, so you get lots of zeros in this matrix. Now, the first component of our model is pretty straightforward. It's just multinomial sampling. So we're going to assume that given a community where we have different taxa with different proportions, we generate a sample of finite size from that just by sampling at random with replacements from the community. So this is very basic stuff. So you've got a probability vector of taxa, which is the community profile, one for each community, which has a probability of observing each species. And then the likelihood then of any given sample of a given size, so I'm denoting the community size here of the nth sample as Jn, then the probability of observing any given sample is just given by a multinomial distribution. So that's very straightforward. I mean, these slides were actually designed for biologists, so for sort of more mathematically inclined people as yourself, I'll go through these bits quite quickly. So, but it should be pretty clear. Okay, the next component to the model is to think about not just having a community profile, but having a prior on that community profile. So we're going to use Bayesian statistics here. And I've got, this slide is basically just a little introduction to the Bayes theorem for people that aren't so familiar with it. But the basic idea behind Bayes' theorem is that the thing we're really interested in is not the likelihood. So this is the likelihood, which is the probability of the data given the parameters. What we're really interested in is the probability of the parameters given the data, the posterior here. And Bayes' theorem is basically that posterior is the product of the likelihood and a prior. The prior represents your belief in the parameters before you even see the data. The denominator there is something called the evidence, and that's just actually it's the numerator of that equation marginalized over all possible parameters. It gives you how much support overall you have for a particular model. The evidence is actually a big sticking point in the application of Bayes' theorem. If it wasn't for the evidence, everything else would be straightforward. The evidence is really, it's a very important quantity because it tells you overall how good your model is, but it's actually very hard to calculate. But anyway, okay. So we then want a prior for our community profiles, okay? And there's a very obvious distribution to use for that, and that's something called the Dirichlet distribution. So the Dirichlet distribution is a distribution over probability vectors. So the probability vectors are the community profiles, and you imagine that this distribution generates many possible community profiles. And the parameter of the Dirichlet distribution I've just denoted there as alpha. And it's a very simple distribution in fact, because basically, so you have one parameter S for each taxa, and that parameter is strictly positive, okay? Which basically means that you've got one more parameter than you do in the community profile itself. So the means of the probability of observing each species is just proportional to alpha S divided by the sum of the alphas. And that sum of the alphas then controls the variance in the distribution. It's actually a precision. So the variance is inversely related to the sum of the alphas. So basically, as we decrease the sum of the alphas, the distributions get more variable, so that's shown there, or we can bias the mean, which is shown there. So the Dirichlet distribution is a very very simple distribution. There's another reason why we want to use it in this context, and that's because it's what we call conjugate to the multinomial. So the key thing about the Dirichlet is that when we use it as a prior for our multinomial, then the posterior distribution is also a Dirichlet distribution. And it's a Dirichlet distribution which is basically just given by, just has parameters which are equal to the original parameters of the Dirichlet plus the observed counts. Okay? So in that sense, the alphas are acting as pseudo counts for the community. It's effectively, so to give you a sort of intuitive understanding, it's like before we start doing our sampling, we imagine putting extra counts already into the sampling boxes. So it acts as a smooth. When we come to calculate the actual community proportions, it's smoothing out the prediction for the community profile according to the counts. So the higher the sum of the alphas, the more smoothing it does, basically. So that's the Dirichlet multinomial. So there we're using the Dirichlet as a prior, but effectively we can take a hierarchical approach and not use it as a prior, but use it as an actual as an actual distribution. So we imagine marginalising over the community profiles to give something called the Dirichlet multinomial distribution. And we need to add a prior on the alpha parameters. So it's sort of like we've gone one level up in the model. Before we were just considering the alphas and the profiles, and the alphas were fixed. Now we allowed the alphas to vary, but we have a prior, we actually use gamma priors, on the alphas themselves. And the point of doing this is it becomes a more flexible distribution than just the multinomial. So we're now using the Dirichlet multinomial distribution to describe our communities. And so this is what we did in our paper in PLOS One. Actually, someone else did exactly the same thing in another PLOS One paper around the same time. But we took a more Bayesian approach. They were basically more frequentist. And this is something we call a plate model. So this describes the model structure. So we've got the observed vectors of counts, the xn. We've got our community profiles, the pn. Both of these are s-dimensional. And we've got n of them, one for each sample. Okay? And then we've got the parameters on the Dirichlet, the alphas, and then we've got the prior, which I've drawn in a square box to represent that it's a fixed parameter on. And this is a very convenient way of writing these probabilistic models. As they get more complicated, it's a very useful way to get track.
What you haven't said anything about the relationship between different PNs.
Relationship between different PNs.
Well,
under the model, the relationship between the different PNs are encapsulated in the Dirichlet. Yeah, because you've got one set of alphas for all the communities. Okay, and then you fit the parameters of the alpha. Okay, so the relationship between the PNs is basically they're being generated by a Dirichlet distribution. This slide tries to intuitively explain that. You're now varying the counts in the Dirichlet prior to try to represent all the communities you observe plus the expected variability in those communities. Okay? That makes sense, I hope? All right. So now we're going to apply it to a real data set, and I'm going to use this data set for most of the analysis in this talk. It's quite old now. It's from 2009. It was a study of twins and their mothers. And I'm not really going to use the feature of the fact that this data is twins in this analysis, but one thing I am going to, do I talk about it? Well, the reason for this study was to look at the effect of obesity on the gut microbiota. So these twins have varying body masses, and it was one of the first studies to look at the effect of body mass on community composition. It was from 16 S genes, V2 samples, 154 different individuals, and yeah, they've been classified into lean, obese, and overweight based on BMI. So we have a lot of reads, over 500,000 reads over these 278 samples. And there's a great variation in sample size from 50 reads to 10,000. So this is a... Are you sniggering, Daniel?
That's well designed.
It's you can't avoid it. It's not designed.
You can always throw out the individual.
Yeah, well, that's what you do in the analysis. I'm just showing this was the raw data. And then, yeah, we classified the reads to the level of the genre using using the RDP database. So the first thing we can do with this is actually just fit a Dirichlet multinomial to the lean and a Dirichlet multinomial to the obese, and use that to describe how lean and obese people differ from each other. And there are some differences. I mean, the nice thing about this is it's a Bayesian approach, so it gives us uncertainties in the prediction. But we'd just about say that there's a higher level of bacteroides in lean than obese people. There's more rosaburia in obese than lean, and more diarrhea than Collinsella. But it's quite a sort of nebulous difference. And this has been one of the problems with actually this question of whether obesity affects the gut microbiota. In actual fact, although they are nebulous, they do seem to be fairly consistent. So we've got a data set we generated from Scottish children just last year, and you actually see the same organisms being over and underrepresented in lean versus obese.
Sorry, so what proportion of lean, for each gender, what proportion of lean children is the direction correct?
Well, I don't know. But yeah, it's a fairly, as I say, it's a fairly nebulous pattern. It's a weak pattern. You can get a sense of how strong it is by asking whether, given the microbiota, you can predict whether somebody is lean or obese. And you can do that with about 75% accuracy. It is quite bad. Yeah, okay. So,
can I ask, do you do a likelihood ratio then on the two alphas?
yes, yeah. In fact, that's exactly what you can do to ask whether the difference overall is significant. I can't remember actually now what result we got. It differs whether you use this frequentist approach or this is a very small part of the talk. Okay? All right. So that was just a comparison of lean and obese. We then wanted to go on and address this question of enterotypes. Now you can do that quite easily within this Dirichlet multinomial model structure. Effectively what you do is you have more than one Dirichlet distribution to describe your whole data. So now our prior on the community profiles PM is actually the sum over k different Dirichlet distributions, each with a different set of alphas and then a different mixing proportion, lambda k, which is basically the prior assumption on the likelihood that the any given community comes from a different Dirichlet component. And this just shows a generation from a two-component Dirichlet mixture, one with a biased mean and one with a symmetric mean.
So you could force the alphas to be 1 or 0.
Pardon?
You could force the alphas to be just 1 or 0.
No, you can't. The alphas have to be strictly positive in a Dirichlet distribution. They can approach 0, but they can't actually be 0. It's actually one of the sort of limit. The assumption of a Dirichlet is that basically everything is possibly there. You can't have a 0 alpha under a Dirichlet assumption. Anyway.
I mean, it's very like the structure mixture model.
It is a mixture model.
Yeah,
it's exactly a mixture
structure model. But you can have a no admixture model and a mix. You can force the. Yeah.
Okay, and then this is just, as I said, this is more for when I was trying to explain this to biologists. This is an intuitive explanation now. So now we're grouping the samples and we're fitting a different Dirichlet prior to the two groups to reflect the different compositions within the groups themselves. Okay, and then this is the plate diagram for the Dirichlet multinomial mixture. So now we've got the community profile. It's being generated by one of k different alphas, and the ZNs is basically the index on the community. So it's which mixture component generated that profile. These were the mixing proportions which are our priors on those ZNs. And then we actually fit the model as a MAP model. It's a maximum a posteriori model. So we basically we use a variant on an EM algorithm to find the most likely set of alpha Ks for the different components, most likely ZNKs. We then, yeah.
Sorry, I'm just trying to understand.
Yeah, that's good.
Each community comes from one of the is it discrete?
Yes, exactly. That's actually a key point about this model.
So it is discrete because you could fit them as a mix of the different.
You could, but that's not what we've done, and it's not actually what the enterotypes problem is about, is it? So the enterotypes is the idea.
It's not clear.
Well, that's more of a philosophical debate. Okay. So one of the key questions you then have is how many mixture components describe the data. Okay. Now, actually, to do this properly, you want that evidence. So the evidence was the overall likelihood of the model. If we could marginalize out the alpha Ks to get the evidence for each different number of components, that would be the correct way to compare one model to another. We can't quite do that, so instead we use something called the Laplace approximation to approximately marginalize out the alphas. And if we do that for the twins data set, we would actually predict that 4 components describe the data best. So this is a bit different to the idea of enterotypes. We're not saying that we've got 4 discrete clusters, and in fact, so this is plotting all the data points on an MNDS plot, which basically approximates their true community distances in 2 dimensions. You can see that they are diffuse components. But yeah, so it is correctly encapsulating this idea of the enterotypes as being sort of modes in the probability space of the community distributions. And this just shows the community profiles for each of the samples in each of the components plus the mean profile. And we get the same basic structure as they observed in the original paper. So one of our components has more Prevotella than the others. I think, that's component 1. One of them is dominated by Bacteroides. One is dominated by Rhinococcus. This component is quite interesting because effectively it's a very high variance component, which isn't dominated by any one individual bacterial type, but has lots of different alternatives, mostly Firmicutes bacterium that could be in it. And the other interesting thing about our analysis compared to the original paper is in the original paper they saw no correlation between the enterotypes and any disease states. We actually find a very significant correlation between enterotype and body mass. So depending on whether you are obese or lean, that affects your probability of deriving from the different enterotypes. So you're much more likely to come from enterotype 2 if you're lean versus obese. So, I mean, we're quite excited about that, but it really, I published it in PLOS ONE a few years ago, didn't get that much attention, but I went on and worked on some more models I'm going to talk about. In the meantime, somebody else took my model, applied it to the HMP data, got a Nature paper, right? Which is slightly annoying in the sense that it makes me think that maybe I should spend less time devising new models and more time just simply using the ones that perhaps I've developed. But yeah, they found very similar things on the HMP. So this is a wider data set now so that they found 4 or 5 Dirichlet components describing the data best, and they found lots of significant correlations between enterotypes and some things that perhaps you wouldn't have expected. They found, for example, that the oral and gut enterotypes were linked together. So if you were more likely to have a Prevotella-dominated oral microbiota, you were more likely to have a Prevotella-dominated gut microbiota, for example. And that wasn't seen in the original HMP analysis where they didn't do this clustering. So yeah, I mean that's a nice successful application of my model in fact.
So the scale on figure A on the y-axis is pretty suspicious here, right?
Yes. Well, I wouldn't say it's suspicious. It points out an important thing, and that's the sort of the number of components isn't really a fixed value. Okay. The correct thing to do with this type of problem is to actually draw the number of components from a distribution. And what I'm going to talk about in a moment could be used for addressing that problem, although I'm using it in a different context. So you could actually use something called a Dirichlet process to define the number of mixing components and then generate a posterior distribution over that.
But if there really were different communities, you would expect orders of magnitude difference in the fit from
it's not.
Well, that's the point. These aren't yes, if you had 3 distinct community types, then yes. But this isn't what the model is trying to encapsulate. It's components within a sort of a space of community distributions.
You can have subtypes.
Yeah.
The biological prior that you should have n distinct. That's it's not a true.
Yeah.
It's like you
So
can have some. Yeah.
this type of modelling isn't supporting the idea that enterotypes are discrete entities. It's supporting the idea that there is shape and structure to the space of community configurations. And then it actually is fairly nebulous exactly how many components correctly describes that structure.
Both from a biological and a statistical point of view, the number of components is the hardest thing to interpret.
Yeah.
Not worth spending too much time.
Well, I don't know. I mean, I think we could. There's something I might return to and try and do a better job on actually. Okay. All right. So that's an example of how we can use this Dirichlet multinomial framework to fit these enterotypes. What I've been doing since then is actually thinking about how to start to model unseen species. So the problem with a Dirichlet distribution is that number of species in it is fixed to the number that you've observed. There's actually something called a Dirichlet process, which is in some sense is the infinite dimensional extension of the Dirichlet distribution, and that naturally encapsulates this uncertainty and the easiest way to understand the Dirichlet process, or what possibly the easiest way, is through something called the Chinese restaurant process. So the Chinese restaurant process is it's a process for generating samples from a Dirichlet process. And the idea is now we've got our community, which is basically a black box, and we pull one sample out of the community, and that will be from a unique type, okay, which I've denoted red here. Then we pull another sample out of the box, and now there's 2 possibilities. We can either see a sample of the same type, so the red type, and now we've got 2 of the first species, or we can see a new species. And under the Dirichlet process, the probability of seeing a new species at each sampling type is proportional to this parameter theta. And the probability that your sample comes from one of the existing species is just proportional to the number of observations of that species you've already seen. So this shows then that, say, for 2 samples, we've got a probability theta over 1 plus theta of observing 2 species, 1 over 1 plus theta of observing 1, and then we can generate a 3rd sample, but now we've got 4 possibilities, basically that corresponding to 1 species, 2 species, and then that's where we've generated 3 species, each sample generating a unique species in turn. Now, the Chinese restaurant process is the easiest way to understand the Dirichlet process, but another way to view it is through something called a stick breaking process. Okay, so in a stick breaking process, we imagine generating, we would generate an infinite number of random variables, which I've denoted eta dash there. And each of those random variables comes from a beta distribution. Okay, so a beta distribution is between 0 and 1, and its parameters are controlled by theta. Okay, and then we generate another set of infinite random variables, the etas, which are basically equal to the eta dashes plus multiplied by by the product of 1 minus all the preceding eta dashes. So the intuitive understanding for this is that we generate our first probability vector by snapping a stick of length 1, okay? And the length of this first break is your first probability component, but then you've got 1 minus that left over. And then the next break of your stick is you break the 1 minus bit. So the key thing about this is you can break the stick an infinite number of times. So you can generate an infinite dimensional distribution under this. And that's also known as the gem distribution, after the names of three people. Probably Alan knows their names. It's well, there's three mathematicians came up with this.
Is one of them.
Yeah. Okay. In actual fact, the Chinese restaurant process is the marginalization of this stick breaking distribution. So the Chinese restaurant process is what you would observe if you integrate out all your gem distributions.
Does this really talk about biology?
No, this bit wasn't. They didn't have this in when I was talking to the biology. Okay, but there's another component to the Dirichlet process, and that's this thing I've denoted H, which is the base distribution. The base distribution can take any form whatsoever. So the Dirichlet process is basically, for each of those infinite number of samples, you draw an additional sample from the base distribution H. So then you imagine having delta functions with weight generated by the stick breaking process at each of your samples H. So I mean that's a little bit, I mean I have to admit I haven't done a good job of explaining that, but perhaps if we get onto the actual application it'll become a little bit clearer. But the key thing is the Dirichlet process has these two components. The theta, which is basically just as for the Dirichlet distribution, controls the dispersion. So the higher the value of theta, the more concentrated around the mean the Dirichlet process is. And then that mean is actually given by another random distribution, this H, which as I said can take any form. All right, so now we're going to get to the biology, which I think Todd talked about, and that's something called Hubble's unified neutral theory of biodiversity. So Hubble's unified neutral theory of biodiversity is actually a stochastic model. It's quite a simple stochastic model. You imagine having you have a mainland island structure. So the mainland is represented by your meta community here, and then you've got each of your local communities is generated by a stochastic process, and so is your meta community. The stochastic process in the meta community is very simple. At each time step, you choose one individual at random, you kill them, and then you have a probability nu of replacing them with a new species, or probability 1 minus nu of replacing them with a species chosen at random within the meta community. When I mean at random, you choose a random individual in the meta community. Okay, so that generates your meta community distribution, and then for your local communities, you have a very similar dynamics. Except for here, when you kill somebody, there's a migration probability that the new individual comes from the meta community, or they're generated from the local community, choosing an individual at random again. And you can probably already see the connection that this might have to a Dirichlet process. But that's basically what Todd showed formally. So what Todd showed formally in his lecture was that Hubble's neutral theory, and in fact not just Hubble's neutral theory, but any neutral model with a mainland island dynamics, a mainland island structure, and a very broad class of neutral dynamics. I think as he explained it, it's as long as it's not possible for one individual to form the majority of the next generation, then this will be true, right? So he showed that effectively we can describe the stationary state of that model by what we call a hierarchical Dirichlet process. Okay, so now each local community, I've denoted Pn, effectively is described by an infinite probability vector, and that probability vector is generated as a Dirichlet process with a dispersion parameter which is just the immigration rate, and a mean which is the meta community distribution. So as you increase the immigration rate, you become more closely focused around the meta community. And then the meta community distribution is just the stick breaking prior, given the biodiversity parameter theta, the gem distribution. So this is a nice observation because it connects ecological theory in a sense with statistical modeling. Statistical modelers have been using hierarchical Dirichlet processes for a while. Ecologists invented the UNTB, and we've actually shown that the two things are equivalent to each other. In fact, a lot of the methods people were using for fitting the UNTB were using this assumption. They just weren't clear about it. The nice thing is that there's a whole machinery for fitting the hierarchical Dirichlet process that we can then use for fitting Hubble's neutral theory of biodiversity. Basically, the strategy we use is something called Gibbs sampling. Okay, so ideally we want to sample from the posterior distribution of the parameters given the data. In this case, the parameters are our biodiversity parameter and the immigration rate. Now, in general, that's a very hard thing to do, but often the conditional distribution, so the posterior of just one parameter, given that you know the other parameters, is a lot more analytically tractable and a lot simpler to sample from. And in fact, in this case, all the conditional distributions are variants on something called the Antionak equation. So did Todd introduce the Antionak equation? Okay, so it's very simple actually. So if we generate T samples from a Dirichlet process, the Antionak equation gives us the probability of observing S different things, given a dispersion parameter theta. And it's basically just proportional to the number of different things we observe, theta to the power S, plus some combination of gamma functions, and then STS. I mean, that's a little bit confusing that I've used S again there, but it's actually the unsigned. It's not so bad. I'm thinking about it's the unsigned Stirling number, in fact. So it's just a coefficient. You don't really need to worry about it. Okay, so that's the Antionak equation. And the Antionak equation enables us to devise this Gibbs sampler for Hubble's model. Okay, so the Gibbs sampler, we basically repeat each of these 4 steps in turn, and we'll end up sampling from the full posterior distribution. The first step, well, actually, the key element to this Gibbs sampler is this variable t. So we introduce big T as what we call an auxiliary variable. So this makes the whole sampling scheme easier. But T actually also has an intuitive understanding. T is for the F species in the nth sample, it's the number of ancestors that generated that population. Because if you think about any given species in a sample, it could have been generated from just a single immigration event, or it could have been generated by up to XNS immigration events. Each individual could have had a separate ancestor from the meta-community. And that auxiliary variable is basically the number of ancestors for each species in each sample. So because the whole model is conjugate, the nice thing is that the meta-community is actually then, if you know these T auxiliary variables, the meta-community is just generated from a Dirichlet distribution where we've got the total number of ancestors generated for each species in the meta-population. So the dot there indicates the sum overall the samples. Then to get the immigration rates, we just basically apply Antionak's equation on the number of ancestors arriving at each site. And similarly at the top level, we apply Antionak's equation again to sample the biodiversity parameter, given the total number of unique species and the total number of ancestors. And then finally, we sample the ancestral states themselves, once again, using the Antionak equation. So yeah, it's basically we use the Antionak equation 3 times and the conjugacy of the Dirichlet and the multinomial once. One thing to note here actually, so the Dirichlet process is an infinite dimensional distribution, but what we're effectively doing there is we've got the unrepresented proportion. So the last thing we draw in that equation, so we've got S plus 1 variables, 1 for each species, plus everything we haven't seen. So it's quite neat actually that the infinite dimensional Dirichlet process is reducing down into a finite dimensional Dirichlet distribution. Okay, so that's how we fit Hubble's model. We also want to test for neutrality. I haven't got a lot of time, so I won't go into this in too much detail, but to test for neutrality, we adapt a method that was used earlier by Rampoletti, and we basically generate lots of, so given that we've fitted the parameters, we generate lots of distributions at random under the assumption of neutrality. We calculate the likelihood of each of those samples, and we compare that likelihood to actual data likelihood. So we're using the likelihood as a sufficient statistic here. But one of the nice things about our model structure is we can do that in one of two ways. We can generate it under the full neutral model, where we use the stick breaking process for the meta community, or we can fix the meta community to be that was fitted in the model. So effectively, we have a test for both local and global neutrality in that sense. Okay, so we tested it out on some simulated data. So the simulation was basically we had a set of islands, I think 50 different islands, and then we allowed immigration from any island to any other, with variable immigration rates for each island. So we have no explicit meta community here. The meta community is being generated from the islands themselves, but we had a gradient of migration probabilities across each island. So the effective immigration rate varied from 2 to 100. And then we generated samples, and then we fitted the UNTB to the resulting samples. And so the dashed line there is the true immigration rate across the samples, and then the solid line was our Bayesian prediction, with the dashed lines giving the Bayesian uncertainties in our prediction. The blue line shows the result of applying an alternative approximate method for fitting the multi-site UNTB. So doing a nice job of reconstructing the immigration rate. What's sort of interesting actually is if you do the test of neutrality, so we would say that this is neutral at the local level, so our p-value is not, pseudo p-value is 0.55, but we'd say that it appeared non-neutral at the meta community level. And that's basically because we didn't have a true mainland island structure. We had this effectively allowing we had a sort of we had an implicit meta population rather than an explicit one. All right, so now we applied it to some real data. So this is a classic data set people use a lot for this stuff. It was 29 locations from forests of tropical trees. And for this data set, we actually would predict that the model, the samples were neutral both at the local and the global levels. But what I've shown here is an NMDS plot of those 29 sites, and the circle size is proportional to the immigration rate. The little black dot is actually the meta population. So you can see that, and then what I did was I found the site that was closest to the meta population in the NMDS plot, and I compared the immigration rate to the distance from that site. You actually found a significant negative correlation. So this is sort of interesting, and this is something one of our reviewers picked up on, and that's that. So we have predicted that the community is neutral, but we're also showing that there is a significant effect of space. So we've used a non-spatial neutral model, but then the immigration rates are then reflecting the spatial structure in the model, which you can view as a good or bad thing. I then applied it to the human gut data that I talked about earlier on, the twin samples. And in that case, we strongly reject neutrality. So the human gut does not appear neutral at all. Another thing raised by our reviewer, though, was that the size of the human gut data set is much larger. Okay? So it may just be that with the small tropical tree data set, we don't have the power to reject neutrality. So what I've shown here is for the human gut, I've randomly subsampled 200 guts, 100 guts, 50 guts, 20 guts. And then I've reduced the sample size, and then I've subsampled down the data, and then I've fitted the neutral model to it. And it's definitely true that your power to reject neutrality is affected by your sample size. It's more strongly affected by the sample number, though. So if you look at when we have just 20 samples, we basically would say that the guts are appearing neutral. And that probably reflects the fact that the major non-neutral signature in these guts is those enterotypes again. So with just 20 samples, we're not picking up that enterotype structure. But anyway, so that was useful, I think, to show how the power of the test depends upon the sample size. And it's actually a big motivation for fitting the multi-site neutral model. Because you can show that if you need lots of sites to be able to properly reject neutrality. Another thing we looked at was subsampling to just specific genre. So rather than fitting the neutral model to the whole data set, I picked out all those individuals from 3 different genera of bacteria, the Bacteroides, the Blautia, and the Faecalibacterium. Here, at the local community level, they appear almost neutral. So for the Blautia, we would say that maybe local community assembly is applying. We would reject it for the other 2. But they appear much more neutral than the whole community structure. And this graph is just showing the metapopulation distributions. So the red line is the fitted metapopulation. The blue line corresponds to the neutral metapopulation. And so this is why it's failing at the whole community level, basically because the meta-community distribution is appearing distinctly non-neutral. Okay, so I'm nearly done actually. So this is just a little bit of analysis I did this afternoon in the hour that was left between when I was meant to be giving my talk and when I actually gave it. So human guts don't appear neutral, but it doesn't mean that fitting the neutral model doesn't tell you something useful. In some sense, the immigration rate is like an estimation of variability for an individual sample. So what I did here was took a different data set that we have, which consists of children with Crohn's disease and children that were healthy. And I fit the neutral model to both of them at the same time, and I found the median immigration rates for the Crohn's disease children and the healthy children. And you can see that there's a very significant difference in immigration rate between the Crohn's and the healthy. Crohn's have a much lower immigration than healthy individuals. And now, there's a lot of difference between Crohn's individuals and healthy individuals. So there's less diversity in Crohn's samples, there's less Shannon's diversity, but in actual fact, the immigration rates gave us a more significant difference than either of those 2 simple measures of diversity. So I mean, that's just a motivation for it as a statistical device, and I think that's probably the direction in which I'm going to go with this research now. So rather than thinking about the neutral model as being a realistic model of the ecology of the organism, because I don't think it is. I don't think at the whole community level guts are neutral. Instead, you can view it as a statistical prior. And this is just to motivate the use of the neutral model or the hierarchical Dirichlet process in this context. So an important question about the obese data sets is whether there's a correlation between body mass and observed diversity. Now, if you look at the raw data, there's a very significant negative correlation. But there's also a strong negative correlation between the sample sizes and body mass. So it just so happens that the majority of the very large samples were from low body mass individuals. Okay?
Wait. It so happens that casts doubt on whether you should be comparing them at all.
Well, I'm going to show how you can deal with that now. Okay, so the standard...
Yes, but no. I mean, if the data had been collected in a different way, then you start to disbelieve that they should really be compared. That's what I'm saying.
All right. Well, just suspend your disbelief for a moment. Okay. So the way people in microbial community analysis would normally deal with this is they would set some limit on sample size, say 1,000. They would throw away all samples that are less than 1,000, and then you subsample everything down to be the same size. The problem with doing that is that you throw away a lot of information. So when I do that for this data set, we don't see a significant correlation between body mass and diversity. But the HDP actually gives us an alternative strategy for equalizing the sample sizes. Rather than subsampling everything down, we can subsample everything up. So we basically fit the HDP to the whole data set. And then we use the model to generate samples under the HDP to basically increase the apparent size of each of the samples. So that's what I've done here. The body mass, so on the y-axis now we have the extrapolated number of species. The x-axis is the body mass. And when we do that, we do find a significant correlation between body mass and diversity. So it's a way to equalise for sample size without throwing things away.
You make it up. I mean, you're...
No, you're not. Okay. Each of the immigration rates effectively defines for each community how likely a new sample is to come from a new species.
That would be true if the model was
And at the top level,
that would be true if the model was true.
right. So I mean,
Can I put this type for questions, right? So we can get Chris finished.
Well, we kind of at the end.
Yeah.
all right. Okay. So to summarise then. So I hope I've motivated Bayesian probabilistic modelling for microbial communities. I demonstrated how we can come up with a clustering model that allows you to have these diffuse enterotypes, variable size samples, and also fit Hubble's UNTB. And then as a statistical device, it's going to allow us to solve this problem of how to account for different sample sizes. Now, to do that properly, we probably actually need to move to what we call a Pitman-Yaw process. So a Pitman-Yaw process is a more flexible Dirichlet process, where you now have two parameters describing the prior rather than just one, and it's got a much longer tail. And I think that's the reason why the standard HDP isn't necessarily fitting the data. Is basically because the real distributions have a fatter tail than in the standard. So this is what I've been working with Todd on over the last two weeks. So he managed to come up with, we came up with Antionak's equation for the Pitman-Yaw process, and then also a Gibbs sampler for it. Okay, and then I've got lots of people to thank for this. Ian Holmes in Berkeley, originally, a discussion with him was where we originally came up with this model structure. Todd Parsons, that does all the analytical mathematics. Leo helps me with the R packages. Keith helped me with the samplers, his former postdoc in Glasgow. The twins data comes from Peter Turnbow, and Umrajaz did a lot of the bioinformatics as well. Okay, thank you. All right, I think, should I stop recording now, or should we do, all right, we'll do a few minutes of questions and I'll stop recording. Any questions?
So where I started getting a bit confused was around, so the Hubble's model is a process, but the data that you have, for the most part, they're not time course data, right? They're one-off.
Yeah, so we're fitting the stationary distribution of Hubble's neutral model across multiple sites.
Okay.
And so, I mean, it's appropriate to compare that to the gut data, where you imagine having multiple sites connected through a metapopulation, but this wouldn't be appropriate for time series data.
And if you had time series data?
Well, actually, I'm talking to somebody at the moment who's got time series data, and I think the first thing to do is to look at the correlation between the data points, because basically you can apply this if you wait long enough such that the time points aren't correlated anymore, then you can treat them as separate samples. That's my guess. But I don't know how, I have no idea. I've told them to calculate the correlation times, and then we'll have a look at the data. I was going to talk to Todd about whether we could use the same strategy to come up with a way of fitting time series data.
Can you just go back a couple of slides,
Mm-hmm.
please?
So what?
Yep, one forward again. Yep. Why is the confidence interval kind of widening out? Is that an artifact of the extrapolation?
Right. The reason for that is basically, as we said, with this data set, it happened to be that the small samples came from high BMI individuals. So this one here is basically a very small sample that has required a lot of extrapolation to take it up to the large sample size. Hence, our uncertainty in the prediction of diversity in that sample is very large. But that's the nice thing, we get an uncertainty in the prediction. So even if you don't believe it, you know exactly how uncertain it is, right?
Well, if the model is true. So,
Right.
so the first thing is that the fact that there's a difference, from an experimental point of view, the fact that there's a difference between the the obese and the non-obese suggests there may be some other difference which may also affect your results. I mean, it's not the process. How many reads come out is it's a reflection of the protocol. So it's kind of suggests there's a difference in protocol between the obese and the non-obese.
Well,
Some-
probably, what it reflects is that the individuals that are obese are more likely to have issues such as an inflamed gut microbiota. Okay, if you've got an inflamed gut microbiota, your sample is going to be worse, in that it's going to have more blood and mucus, etc. in it, and then that may affect the library preparation process. So yeah, that would be the sort
But
of.
that would also affect the species diversity that you might.
No, I think that's a real phenomenon actually. But
No, it may be a real phenomenon, but it's going to affect. It'll affect the diversity that you estimate. I mean,
yeah, of course it will. So you're saying the diversity will affect the diversity that we estimate?
No, I'm just saying that the fact that you have few reads.
Yeah,
So-
well, that affects the observed diversity. Exactly, that's the whole point of doing this analysis.
Yeah, but
You've got an explicit multinomial sampling. Okay, so you anyway. All right, I think we'll
Yeah.
sort of stop recording now.